When we think of structures keeping us alive, what springs to our minds? Our heart? Our lungs? Our liver? However, today I'm going to talk about a small structure present inside each of our cells which works tirelessly to keep us alive and functioning every day. The mitochondria. So today I'm going to talk about my research, which is about studying mitochondrial proteins in animals. And this is the outline of my talk. What are mitochondria and why are they important? What do we know about mitochondrial proteins? What animals do I work with? How do I identify and study mitochondrial proteins? And what did I find out from my research? So what are mitochondria and why are they important? Inside each of our cells, there are many organelles. Undoubtedly, one of the most important is the mitochondria. How these mitochondria ended up in our cells is fascinating. Over 1 billion years ago, these mitochondria were actually free-living bacterial cells. Eventually, they were taken up by a much bigger cell. They were very important for this big cell and hence mitochondria were maintained as organelles inside of these big cells, which eventually became the cells which make up our body. So, why are mitochondria so important? Mitochondria are called as the powerhouses of the cell. This is because they generate almost all the energy our cell needs for various functions. They are also involved in many other important cellular functions such as cell death and aging. In fact, defects in mitochondrial functions have been linked to diseases like cancer, Huntington's, Parkinson's, and even Alzheimer's. A recent survey by the National Institute of Health projected that the number of Americans with Alzheimer's will more than double by the year 2060. Now, all this important work inside of the mitochondria is carried out by proteins. In human mitochondria, there are over 1600 proteins carrying out all these important functions. My research goal is to study this set of mitochondrial proteins in animals. So, what kind of animals do I work with? Animals can be broadly classified into two groups. Bilaterian animals, including us, who have bilateral symmetry, that is, only one plane dividing our body into two halves. And the other group is a non-bilaterian animal. These include animals like sponges, corals, jellyfishes, sea walnuts, sea anemones, and many more. These have no bilateral symmetry. Mitochondrial proteins have been very well studied in some bilaterian animals like humans and mice. However, no such study exists in non-bilaterian animals. My work focuses on this group of non-bilaterian animals. So, how do I identify and study these mitochondrial proteins? In my project, I use publicly available data and use statistical and computational techniques to identify mitochondrial proteins and characterize their functions. The overall goal is to provide the first comprehensive review of mitochondrial proteins in non-bilaterian animals, and then use this data to see how important mitochondrial functions and pathways have evolved in animals. So, additionally, these proteins in mitochondria are actually synthesized outside the mitochondria and they have to be targeted into this organelle. And the way they are targeted is via a very specific signal present inside each of them. In my research, I also identified these targeting signals in mitochondrial proteins and analyzed them. So, what did I find out from my studies? Our analysis showed that these sets of mitochondrial proteins in non-bilaterian animals are extremely diverse with respect to both size and content. Some sponges have more than 700 more proteins in their mitochondria than we do in our mitochondria, and some non-bilaterian animals have less than half the number of mitochondrial proteins. Some mitochondrial pathways were found in the mitochondria of all non-bilaterian animals, whereas proteins involved in mitochondrial functions like cell death, were not identified in many non-bilateral animals. Additionally, we also found that these signals which target mitochondrial proteins to the mitochondria differed between non-bilateral animals like the sponges and the jellyfishes and humans. It is surprising how little we know about the set of proteins in an organelle so important for our survival. So in conclusion, while these are just the initial steps, we can learn a lot about the evolution of mitochondria from these diverse groups of animals.